So today we're going to talk about those three concepts that we started talking about last lesson, which is about those things that are genomes, um, how genes fit into that, and then how alleles then fit into genes. Okay, very important concept that has some big implications, such as here with Starbuck, which is a famous Canadian bull whose genome was so desirable that his sperm actually sold for $25 million during his life, and he actually has more than 200,000 daughters. All right, pause the video here and have a go at the three quick questions. All right, so for question one, it's asking you name one positive and one negative about sexual and asexual reproduction. Okay, remember that what's positive for one is going to be negative for the other one. So let's talk about just the positives. So a sexual reproduction positive is increased genetic diversity or genetic variation, all right? And then from that, we can actually get a bunch of other positives, such as the fact that it's an organism that's a sexually reproducing organism is actually going to be better suited to environmental changes, or it's going to be more likely to be able to survive diseases, okay? A bunch of other positives of asexual reproduction, which is things like being fast, low energy, okay, no need for a mate, lots and lots of examples. Question 2 says, although independent assortment leads to a huge number of possible human variation in our sex cells, crossing over brings that number up to nearly infinity. Let's explain the statement, okay? So it's quite simple. How I think of it is that independent assortment is like a reshuffling. Okay, so if you took a deck of cards and you took that deck of cards and they were completely in order, both in number and in the suit, you could take around that deck of cards and shuffle it up, and then it would be out of order. And then you could shuffle up again and it would be out of order in a different way. But if you actually did that 100,000 times, you'd eventually actually get to an order that you've gotten already. Okay, and that's how independent assortment works. We have 23 pairs of chromosomes that can be decided which order they go in completely randomly by independent assortment, but we're always going to just be able to use what's available to us. Crossing over, on the other hand, right, actually creates new chromosomal variations. So we're going to get new types of chromosomes from crossing over. Okay, question 3 says, name the 5 stages of the cell cycle denoted by the letters S, G2, G1, C, and M. All right, so we have synthesis, gap 2, gap 1, cytokinesis, and mitosis. Okay, and if we're going to place them in a logical order, I like starting over here with G1, and then we're going to go synthesis, so gap 1, synthesis, gap 2, mitosis, cytokinesis, and then back to gap 1 because we're talking about a cycle. Okay, so in the study design, you guys have to know about Unit 2, Area Study 1, genomes, genes, and alleles. We have to distinguish those 3 words. Okay, define and distinguish them. Remember that when we're talking about differences, we have to talk about the difference between the 2 things. And then we're going to branch out into the genome a little bit more as pretty much a good definition, the sum total of an organism's DNA measured in the number of base pairs contained in a haploid set of chromosomes. Great definition for that word, genome. All right, so to quote Dr. Seuss, what makes you you? Okay, so today you are you. That is truer than true. There is no one alive who is youer than you. Okay, and genetics takes this on, okay, because DNA is going to be specific to every single person and every single living thing out there. Okay, and let's talk about this thing DNA. DNA is deoxyribonucleic acid, and this is a biomacromolecule, which means that it's made up of atoms, all right, and it's big. That's what macro means. And it's to do with life. It's essential for life. All right. And it's a biomacromolecule that carries the genetic code for protein expression. So, genetic code for a protein. And this dictates essentially all aspects of life. Okay. So, we can see in this little diagram, we have the DNA, which makes RNA, and that makes a specific protein. DNA can also be represented like this, and we can look at this representation in two components. We have the phosphate deoxyribose backbone, or I like just calling it the sugar phosphate backbone. So this is the deoxyribose component, and this is the phosphate component, and that makes up this backbone, and we have it going one way on one side, so it goes this way on this side, and then on the other side of the DNA, it goes that way, so we call it anti-parallel. Okay, and the outside of the DNA, the backbone, it's doing is just protecting and making sure that it's structurally sound. Okay, what's really important about the DNA is these four guys: adenine, thymine, guanine, and cytosine, or the nitrogenous or N bases. And why they're important is because the nitrogenous base sequence of DNA is what actually results in the variety of living things in the world. Okay? And the variety comes from these guys, the genes. All right. So, what is a gene? A gene is the basic functional unit of heredity. Okay. It's how we pretty much measure the different potential proteins that we have. Okay. And what a gene is made up of, it's made up of a specific section of DNA. So, for example, this gene is this specific section of the DNA. Okay. And so, if we unwire the chromosomes, which are in the nucleus of a cell, we're going to pull out the DNA and it's going to be full of these genes. And a specific section of DNA which codes for a specific protein is what we call a gene. In humans, these genes can actually vary from about a couple of hundred DNA base pairs to more than 2 million base pairs. All right. So they can go from smallish to gigantic. And within a species, 99.9% of our DNA is actually the exact same as the person next to us, which is pretty nuts. Turn around, look at the person next to you. You don't think they are alike to you at all. So, where is the variation? And because it makes sense that we're so genetically alike. Check this out, right? As much as these two young babies, look very, very different, right, in skin color, hair color, different physical features, these guys are a lot more alike. They are 99.9% alike. But compared to a chimp, this little guy down the bottom, they're not as alike. They're actually only 99% like the chimp, which is still pretty close. Well, not 99.99%, it's more like 98.7%, which is still pretty close, but it's certainly not as close as this number up here. So the variation in genes actually comes from changes in those nitrogenous base sequences of the DNA making up the gene. Okay, so pretty much the changes in the A, T, C's, and G's. And that leads to different variations of genes. And we call these different variations alleles. Okay, a great example of allele is widow's peak. We love talking about widow's peak because it's just coded by one single simple genetics gene. All right, and so if you have widow's peak, we say that you have the big W allele. Okay, if you don't have widow's peak, you actually have the little W alleles. So they're just different versions of the gene that decides what your hair looks like right here. As you can see, I have widow's peak. All right, so let's look at a specific case study, one of the most important case studies, which is the ABO blood groups. Okay, the ABO blood group gene codes for a type of protein which actually determines an individual's blood type. For example, I there it is. Went and gave blood, and then I found out that I have A positive blood. okay? And that's my blood typing, okay? So this gene is actually about, well, pretty much 20,038 base pairs long. If you ever see BP, by the way, that's the N base pairs, the length, okay? And it's found on chromosome 9, specifically this part right here. So this is all chromosome 9, specifically right there, which is called the locus. Okay, that's the location of that gene. Okay, so there's three allelic variations. There's three different alleles for the ABO gene. Okay, we have the A allele, the B allele, and the O allele. And pretty much what these decide is that if you have an A allele, you're going to have type A red blood cells. Okay, you have this marker, that's the A marker. If you have the B allele, you're going to have the type B red blood cells, having the B marker. If you have the O allele, it actually means that you don't have a functional gene, you don't get that protein made, and so you have absolutely no markers. Okay? I might just move myself over there. This then leads to the fourth variety, okay, because the ABO blood grouping works in a slightly more complex genetic way, in that if you have both the A and the B, okay, so you have the AB genotype, you actually have AB blood. Okay, you have both the two antigens, the two markers on the surface of your red blood cells. So let's compare the nucleotide sequence of these three different alleles. Okay, so with these ABO alleles, let's call, just for convenience, there's no normal, all right, but just for convenience, let's say that this specific type of A allele, all right, is the normal. Okay, it's the new nitrogenous base sequence that we're going to call normal. Okay, and so we read it just going down this way. All right, so they have guanine and then adenine and then cytosine and so on and so on and so on. All right, and this is a specific spot of the gene. Obviously, we said the gene was 20,000 base pairs long. Okay, this is only spot number 261 to 1060. So there's even stuff missing in between. But so we're just looking at specific A, B, A, T, C, and G's that's on that gene. Okay. We can change through mutations these A, T, C's, and G's, these nitrogenous bases. So for example, if we get a change at spot 646, this thymine becoming an adenine, nothing happens. We still have the A allele. Our body is really good at recognizing small changes, and it usually a change is bad, so it doesn't like changing. But if we changed the G allele, the G nitrogenous base, at spot 703 to an adenine, so the guanine to an adenine, we're one step closer to getting the B allele, so the B marker, okay? But if you notice, we actually need a lot more changes to happen. So how many changes actually need to happen to change the marker? So if all of these are the A marker and we wanted to change to the B marker, let's have a look. We had to change 1, 2, 3, 4, 5, 6, 7 here to make a change. Okay, so to go from A to B, you need at least 7 changes in your nitrogenous bases. All right, A to O, on the other hand, if you look here, there's a few times that we need a bunch of changes. So these are the O's. There's a few times we need a bunch of changes. So here, 1, 2, 3, 4, 5, 6 changes. But simply if we were to change this guanine at the start to this little triangle just means anything else, okay, so either an A, T, or a C, we're actually automatically going to change to an O, okay? So the number of changes that need to occur, sometimes they're really small, sometimes there's lots and lots of changes, sometimes you have to change whole chunks of the DNA, okay? If we were to think of what proportion of the total gene this is, we're actually still talking about a very small proportion. We said that the ABO blood group gene is about 20,000 base pairs long, okay, this is only 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17 base pairs. We're only talking about 17 base pairs out of that total 20,000. All right, so it's a small, tiny changes, and it shows why we're so alike to our neighbor, but if we have tiny little changes in our base pairs, we can have potential differences which make a huge difference. These ABO blood groupings decides whether you can have a blood transfusion from someone but not from someone else. All right, so now let's talk about the genome. Okay, so we've talked about genes and we've talked about alleles and how they make up genes. We'll go into that a bit more when we talk about actual inheritance and how our genes make up us. But a genome is pretty much the organism's complete set of DNA. So it includes all of its genes. Okay, each genome contains all of the information needed to build and maintain that organism. Okay, it's all coded inside of your genome. And in eukaryotes, like us and like trees and like mushrooms and like the tardigrade, okay, a copy of the entire genome, so more than 3 billion base pairs, is found in all of the cells that have a nucleus. So nearly all of the cells of our bodies. All of this genomic information of an organism is actually contained in one haploid cell of chromosome. So we know that, so this is what we call a karyotype. It's a very complex karyotype, but it is a karyotype. And you can see that we're just going through and showing all of the human chromosomes. So we have chromosome 1, 2, all the way to 22. But then we have the exception that we need both sex chromosomes, especially if you're talking about a male, because the two sex chromosomes are different, okay? And we can actually compare genomes of organisms, okay? And this gives us a lot of information. So, for example, have a look at this table. Within this table, which organism has the biggest genome? Well, it depends, okay? Because if you're talking about just simply the number of base pairs, simply the number of base pairs, so the nitrogenous bases, then us humans, we have the biggest genome in this table. All right, and if you talk about chromosome number, us humans still have the biggest genome. But if you go on to talking about the number of genes, we have a lot. We have 21,000 genes, but this weed, the Arabidopsis, actually has the biggest genome in terms of number of genes. Okay, and so this raises the question about what's the relationship between the complexity and the genome size. So we're obviously very complex organisms, or we like to think we are at least, and that's evident in terms of our genome sizes. But if you look at something like a fruit fly, which is an animal, all right, they only have 8 chromosomes, and compared to us, a smaller genome, okay? But then you compare it to a weed, all right? They have a much bigger genome. So there is a relationship. Yes, there is a relationship, but it's not a hard fact. Not just because we have more genes is it more complex, okay? An interesting two organisms in here that I've included is E. coli bacteria and HIV, which is the virus that causes AIDS, okay? It's interesting to see that the E. coli, as much as it's the simplest different living thing that can exist, all right, it's still made up of 4.6 million base pairs, okay? But those 4.6 million base pairs, it only has 3,000 or so genes, okay? Remember, it's all in one chromosome because it has a circular chromosome. Viruses, on the other hand, which is not living, okay, they still have DNA, which is a bit mind-blowing. Well, actually, HIV virus doesn't have RNA. It only has RNA, which is a little brother to DNA. But they only have about 9,700 base pairs and only 9 genes to make that huge mess that they make.